Hi, how are you? I am ready for chapter 11. I hope you're too. So, now that we already classify our microbes into two domains, three domains, right? That are microbes that are eukaryotes. So, we have bacteria, archaea, and eukarya domain. And in five kingdoms, no, sorry, I wanted to clarify that. In four kingdoms. I think I said five kingdoms in the previous video. Clarifying there's not five kingdoms, it's four kingdoms. The bacteria and archaea do not belong to a kingdom. Prokaryotes is not a kingdom anymore. It used to be, okay? So our new book clarified that these bacteria and archaea just have a domain, and then under domain you're gonna go straight to phylum, okay? Um, the kingdoms are just the ones inside the eukarya, which are animalia, plant, fungi, and protist. Clear? Hope so. Okay, guys, so now that we have this clear, we're going to talk about two domains today, which are the domain bacteria or eubacteria and archaea. Archaea is super quick. We're going to talk about that for like 1 minute, and the rest of the time we're going to spend talking about bacteria. So before I even start, the first thing I want to show you is the learning objectives as usual and something else. So my learning objectives for chapter 11 say gram-negative bacteria, which are the protobacteria and the non-proteobacteria. I don't need you to know who is proteo and who is non-proteobacteria, okay? I just need you to know who is gram-negative and who is gram-positive. And then get gram-positive bacteria. So for all these groups, gram-positive and gram-negative, know the genera, the relevant features. You don't need to know the families because sometimes I mention the family, but you don't need to know that. I'm not going to ask you about families and groups where they belong. So how are we going to study this? And then I say here, see tables posted in course materials. So if you go on this week's folder, you're going to see a Word file. And that file is this file. Let me zoom it out so you can see the whole thing. Okay. So what is this? This is a table that I'm giving to you to fill. So it says here, these are all the protobacteria gram-negative and the non-protobacteria gram-negative. So this first table is just gram-negative bacteria. The genera are here in this column, okay? So what I told you that you don't need to know are these families here. I don't need you to know these families. The only family that I'm really gonna highlight for you here is this one, Enterobacteriaceae. Okay, because they are the gut microbiome, so I want you guys to have a better understanding of all the bacteria that belong to this group. So clear, you know Enterobacteriaceae, you know the genera, you know relevant features that we're going to talk about now, and you're going to, as you watch my video, please have this table printed and fill it up as you're watching, okay? And then diseases if it's a microbe that causes disease, okay? So one thing I want to also then continuing, if we keep looking at this table, you have the other part, which is the gram-positives. So here again, you don't need to know the families. There are three families for gram-positive, but you need to know the genera, and you need to know relevant features for this bacteria and if they cause disease. I wanted to mention that we're going to mention the diseases, but we're not going to go deep into the disease. And you may be like, what is that disease? What is, for example, I wrote here endocarditis. I just gave you a tip here. What is that? And I'm just moving on to the next thing, because we are going to talk from chapter 21, actually 19, and then 21 through 26, only about these diseases. So anything that we're learning now, you're going to see how the course gets a little repetitive, but when I repeat something that you already know, I'm adding information to it. So it's like you're having a small knowledge that I keep adding and adding and adding to that little part until it gets much bigger. Okay, that's what we're doing here. So I want you guys to have that in mind. So if you read about a name of a disease and you're like, what is that? It is something that you're going to learn later. But the more you get familiar with these names of microbes and of diseases and their features, the more comfortable you're going to feel later. That's why I'm already introducing these diseases. Clear? Okay. If everything is clear, let's move on. The book has a table that shows the classification of bacteria into gram-positive, into gram-negative, and into archaea, which is not bacteria, right? Prokaryotes in general. And here it shows some classes and some features and some phyla. I'm just moving forward from this table because we're going to talk about them one by one, okay? So the Proteobacteria is a very, very big group composed of 5 classes. They're all gram-negative and they're all chemoheterotrophic, okay? So what does that mean here? All these bacteria use carbon source from a chemical and energy from a chemical and don't produce their own food. They don't do anabolism of carbohydrates, only catabolism, okay? That's what chemoheterotrophic means, and I know you know that already at this point. They are able to grow, so now we divide the Proteobacteria into alpha, beta, gamma, epsilon, and yeah, that's it. So again, you don't need to know this class. But inside the alpha Proteobacteria, you have the rhizobium. So this is one bacteria that is in our table. So the rhizobium is not a bacterium that causes any disease. The rhizobium is a bacteria that likes to grow on roots of trees. So it's a symbiont. This bacteria is able to fix nitrogen in plants. And what does that mean? That means that the nitrogen that is in the air gets captured by this bacteria, so the nitrogen gas is transformed into nitrate and nitrate. And then the plant can absorb that, and we can absorb because we eat the plant, hopefully, right? So that's what that means. So that's the rhizobium, fixing nitrogen and grows on plants' roots. Now let's go up in our table and talk about the Rickettsia. The Rickettsia is a genus that is obligate intracellular parasite. What does that mean? It means that Rickettsias cannot multiply inside cells. Okay? They need to be inside a host cell to survive and grow. They cause many diseases that are considered spotted fevers. So it's fevers that have like very high temperature and specific features. For example, Rocky Mountain spotted fever, okay? And typhus is another type of disease that they cause. They also need an arthropod, like an insect or a tick, for transmission, okay? So this is just showing in this picture a rickettsia, these little dots inside a chicken embryo cell, and masses of rickettsia in the nucleus, and this is just showing the bacteria here. So this layer around the bacteria is a slime layer. Remember, that gives them protection. It's like a glycocalyx layer that allows them to be protected against phagocytosis, for example. And then, still in the alpha Proteobacteria, we have the Bartonella, that is a human pathogen and causes cat scratch disease. Bartonella is a bacillus. And then the Brucella, which is another obligate parasite of mammals, so cannot grow outside of cell. and they survive phagocytosis. They have this ability of, skipping the phagocytosis and not dying through that process. So they, for example, enter a macrophage and the macrophage cannot kill them. Okay? The disease that they cause is called brucellosis. Again, we're going to talk about that a lot more later. Nitrobacter and Nitrosomonas. Nitrobacter is a gram-negative, but Nitrosomonas is a gram-positive, just so you don't get confused, because we're talking now all about gram-negatives, right? The first table we're filling is all gram-negatives. By the way, what does it mean to be a gram-negative bacterium? Think about that. What does it mean to be gram-positive bacteria? What can you tell about their cell wall? Make sure you know the answer to these questions, okay? So these bacteria are chemoautotrophic, so they can use a chemical as a source of energy. That's usually an inorganic chemical. I said something about carbon, but the source of energy is always the inorganic molecule. And then the organic molecule is going to be the source of carbon, right? And they're autotrophic, meaning they can fix nitrogen. They can use a molecule that is composed of nitrogen to make one that is absorbable by plants and not toxic. Because remember, ammonia is toxic, right? So they transform the ammonia in the soil into something that we can and the plants can absorb. So that's their function. Do not cause disease. Spirillum is another free living bacterium found in fresh water. And they're aerobe and they move by flagella. Okay? Bucholderia. The Bucholderia degrades a lot of different molecules. They have like a very diverse metabolism. This type of organisms that just can eat anything, they can survive on anything. Okay? They degrade and use as source of energy. And then we have a Burkholderia that causes melioidosis. Bordetella. This is the bacteria that causes whooping cough. They're non-motile, but they grab the cilia of your trachea. Remember, we have cilia in the trachea. They grab that and they destroy the ciliated cells and the cilia. And that's what causes that crazy cough, because you use the cilia to brush the mucus to your esophagus and trapping microbes and take all that out of your airway. If you don't have the cilia, you're going to try to cough to eliminate that mucus, but it's not a cough that is going to lead to a resolution, okay, because your cilia are gone because of the Bordetella infection. There is one type that also causes, I know because I remember my dog got that vaccine, that causes kennel cough in dogs, okay? This is a spirillum. You see how they have this flagella amphitrichus. Let me show you here. On both sides. Remember how this is the amphitrichus flagella, and they're a spiral-shaped bacteria. Now we're going to talk about Neisseria. So now we're talking about beta, right? So Neisseria can mostly cause gonorrhea and meningococcal meningitis. And Neisseria, look at that. They're diplococci. It's two cocci together in both cases, the meningitis and the gonorrhea. The difference is that the gonorrhea has a lot of pili. You see all these little hairs, and that's not what you see in the meningitis. So these two bacteria are actually not these two bacteria. The meningitis is present in the microbiome of your throat, and so you have the bacteria in your body. Most of the people do, and then they can become pathogenic. Okay, we're gonna talk about meningitis a lot more later. They're all aerobes. That's why they like the airway. And they also can grow in mucous membranes, which is the case of the Neisseria gonorrhea. this is what Neisseria gonorrhea do. They cause something that is called membrane ruffling, which is they induce the cytoskeleton of the host cell to make like pseudopods so they can dig inside the cell. That kind of helps them to move in. So they manipulate the host to kind of hug them and then they go in. Okay? So that's super tricky. This bacteria is actually not in our list. Francisella is one of the gamma protobacteria, and it is, yeah, it's not here. And it's transmitted by tick, and it causes a disease. These are tick present in the rabbit and in deer, and it causes tularemia. But we can contract from these animals and from tick bites, tularemia. Okay, still in the gamma protobacteria, now we have the family Pseudomonales. One of the main genera of this group is the Pseudomonas. Pseudomonas is an anaerobic bacteria that is an opportunistic pathogen. I think it's the first time we see this word here. So let's define what is an opportunistic pathogen. It's a pathogen that can already be part of your microbiota, can be already living in your body and not causing any disease. And if your immune system drops the effectiveness, what happens? The effectiveness, it causes disease. It becomes pathogenic. Okay? So you can have Pseudomonas in your body and it's not going to do anything. And one day they cause disease. So usually this type of opportunistic disease that they cause are urinary infection, and they tend to infect wounds, mostly the ones caused by burn. Okay, so look, it has a polar flagellum, actually two. See here, you see two flagella, and some pili around them. So that's the Pseudomonas. The Pseudomonas aeruginosa is the one that we usually see causing. It's a green bacteria. You know, when you see pus in an infection in a wound and that pus gets green, that's the pyocyanin from the Pseudomonas. It's a pigment that is green. Okay? Other from the same Pseudomonales family are Moraxella, that causes conjunctivitis, and Acinetobacter, that causes a respiratory disease and is also very resistant to antibiotics. You find Acinetobacter a lot in soil and in water. Okay? These are all strictly anaerobe bacteria. Okay? They cannot live in the absence of oxygen. All the Pseudomonales. We're going to use the Pseudomonas in the lab as an example of an aerobic bacteria. Okay, so I'm going to stop here and then we're going to continue talking about gamma protobacteria in the next session and the non-protobacteria in just a minute. See you soon.